call this lecture or this story the apartment in the mansion story. And this idea is that the prokaryote and eukaryotic cells that we've been talking about many times already are different because not so much their size, there's certainly a size difference, but because this eukaryotic cell, this big one here, has these little compartments, all right? And so there's a term we need to learn real quick, and that is compartmentalization. That's what this story is really all about. It's about the fact that there are essentially little compartments within that big cell where you can separate chemical function. And the little cell doesn't have this, all right? So the bacteria over here doesn't have compartments. This large cell does have these compartments, and you can basically do some fancy chemistry in there, all right? Now that fancy chemistry that is going on in these compartments, all right, that we call organelles, are sometimes harsh, really nasty, like you're using acids and stuff like that normally would damage the cell, but because it's kept inside these compartments, it doesn't, and hopefully it doesn't leak, right? It doesn't damage the other parts of the cell, and specifically the DNA. Now the DNA is compartmentalized inside the nucleus, all right, the nucleus, and the DNA is in here. The DNA has to be kept very safe, so it gets its own compartment, and everything having to do with DNA is typically done in the nucleus where it can be kept safe, all right? That's an important part of this lecture, is for you to understand that compartmentalization, one of the primary functions of it is to protect the DNA. Now over here in this little bacteria, the DNA here is in blue, and notice it says no nucleus. There is no nucleus here to protect the DNA. So what do you think this does? Well, essentially it limits the chemical abilities of the bacteria, because it can't do anything too harsh. It can't digest certain things to use them for energy or to kind of like to build itself up, to create new structures. It can't digest the food that this large one can, because it can't create the diverse chemistry that this one can. If this one brings something into the cell, all right, to digest it, it can stick it in one of these compartments and use all kinds of fancy chemistry to digest it into the pieces it needs to function. Whereas the bacteria is stuck with just whatever it can do within its whole cell body. We call this region in between the organelles the cytosol, or the cytoplasm. You have cytoplasm here, but other places you'll call it the cytosol. And over here, the cytoplasm, the DNA is just sitting in the cytoplasm. It doesn't have its own compartment. And so therefore, anything that you do out here is going to affect the DNA, and that limits what you can do over here in the little bacteria. It does not limit what you can do over here, because the DNA has its own compartment. So this concept of compartmentalization is really important, and it's one of the major distinctions between the bacteria and the prokaryotic cell structure and the eukaryotic cell structure. We've talked about the fact that prokaryotes have archaea and bacteria in them now, but the idea is both of them use this structure. They don't have organelles, or maybe they have some rudimentary vacuoles, but they don't have anything protecting the DNA. And this is one of the key deciding factors on these being two different domains, organelles and protecting the DNA. This one gets to eat a lot more different things, It has a lot more chemical functions. It needs more DNA to provide information for creating these, and it needs to protect that DNA. So this is a much more complex organism than this one. This one has maybe 3,000 genes, and over here maybe you have 3 million genes. 30,000 genes. So 3,000 to 30,000, that's 10 times more genes and far more, we'll talk about it at the end of the course, far more regulation of the genes, which may be more important than the genes themselves. So there's a lot going on over here, especially chemistry-wise, a lot of different chemicals moving through here. It can eat a lot of different things. It can process a lot of things and turn one thing into another thing chemically. Bacteria can't do a lot of that. But that's one of the main differences. This cell can react chemically to its environment. This one is very limited, and so it limits the environment that it can live in. Bacteria tend to survive, but they don't thrive except for in very specific cases, whereas our cells can pretty much survive because they have lots of chemical abilities, and they specialize in a different way. And we'll talk about that when we talk about development. Alright, so here are some of the things that they have in common right here. And for this picture, I'm not as worried about you knowing this over here, but I do want you to know what prokaryotic and eukaryotic cells have in common. They do have a plasma membrane. That's the outside membrane separating them from their environment. And that is the same membrane that we use to create the organelles, these little organs, these little places where you do all this fancy chemistry. Alright, they all have ribosomes. That's necessary for all life. All living things have ribosomes. Now, chromosomes. Here's where I disagree with this book or this picture. A chromosome is supposed to be linear, and it also has something called histones, and it wraps up. It's really a slang term to call the DNA over here a chromosome. They call it the bacterial chromosome or the prokaryotic chromosome, but it's not, because it's a loop. It's a closed loop, so it's not linear. It doesn't have these certain structures that let it form true chromosomes called histones, and we'll talk about that structure later. So this is definitely not a chromosome. The real technical term is a genophore. I won't test you on that. The slang term all the way up, even when you get into graduate school, will always be the bacterial chromosome, but that is technically not a correct term. It's just a slang term, and a lot of people don't even realize that. People with PhDs don't realize that that's not a chromosome. And I'm not being nitpicky here. It's just that a chromosome has very distinct structures, and when you tell someone this, you gotta go, oh, that's right. Okay, yeah, that's not a chromosome, is it? Yes, you call this a genophore, whereas over here you'll have chromosomes. That the DNA, those are structures that the DNA organizes itself in, and we call those chromosomes, and we'll talk a lot about that later. Alright, let's see here. I think that's it for the pictures there. Let's talk about this little story in the organelles. So the organelles are like rooms in a house. And so this story goes, I call it the apartment mansion story, and we think about the rooms of the house as the organelles. And if you have a big house, all right, like a mansion, all right, like our eukaryote here, this is the mansion, all right, and this is going to be our little apartment, a little efficiency apartment. Now, if you think about this, okay, the rooms themselves are made out of what we call membrane, and we'll talk about the structure of the membrane later, but they make up the walls of the house. They make up the walls on the outside of the house, whether it's a mansion or a little apartment, and the walls are meant in a house, just like the cells are meant to keep things separated. You do certain functions within these rooms, within these organelles, and so some are designed for digestion and some are designed for protecting the DNA. Well, in a house, it's for doing different functions as well. You have things like cooking, you do in the kitchen, you watch TV in the living room, you sleep in the bedroom, and you separate these so that usually they don't interfere with one another. Just like in the cells. One of the limitations of the bacteria is that it's limited to what it can do chemically. And if you think of it that way, if you live in a house, if you live in a little efficiency, all right, and let's say there are 5 people in there. Well, 5 people living in that little efficiency apartment are going to get on each other's nerves. They're not going to do anything very well because other people are doing things at the same time. So if you're cooking or watching TV, well, that makes noise and smells and things, and it'll interfere with people sleeping, or maybe the frying pan interferes with you hearing the TV. Say you do have an extra room, say a bathroom. Well, even then, okay, you take a shower. Well, you can't come out of the shower without, putting on a robe or something or dressing. You have to do it in the shower. You can't go move around and do anything as you like because there are other people out there. All right, so even the efficiency is not good if you're trying to do more than one thing at a time. All right. The mansion, though, on the other hand, you might have 10 bedrooms, and you might have 2 kitchens. People want to do things at the same time. There may be people living in a mansion and they never meet each other. There are so many, you don't even have to use the same kitchen. Certainly don't have to use the same bathroom. There might be 3 bedrooms to choose from, and might have its own movie room. So a mansion, people can do things and not interfere with one another. And so the idea of using this analogy is the same here. By separating and isolating where you do things, what's going on, you don't interfere with other things, and you can get more done. And that's exactly what happens with the cells. The little bacteria cannot separate its chemical functions into different compartments, and so those chemical functions can interfere with each other. Or if you were trying to do something difficult, like say digest something, you might damage the DNA or some other part of the cell. You can't do that, and so therefore you're limited in what you can do as a bacteria, just as you are limited in what you can do if you've got several people living in a little apartment. If they're living in a mansion, like the eukaryotic cell, they can do all kinds of things. They do not interfere with each other chemically if you've got compartments, if you've got rooms. All right, the rooms in the mansion are like the organelles, like the compartments in the cell. If you have a lot of them, you can do a lot of diverse chemistry. If you don't have any at all, you can really only do kind of one thing at a time, and you're really limited on what you can do. So, because you might damage the internal structure. So because of this, I call this the apartment mansion story, and it's all about why compartmentalization is so important, and that is the structure of organelles. And it's truly the reason our cells are so different. You can put different organelles in there. A little apartment is pretty much generic. It's got one room, and you put pretty much the same things in all of them. You put a bed, maybe you have a bathroom on the side, but you have a bed, a kitchen, you put a TV in there. Everything's in the one room. All of these apartments, if you go to a hotel room or something, they all have pretty much the same thing in them. But you go to somebody's house, especially a mansion, and you've got all kinds of things going on. You'll have different things in there, in different rooms and different functions, and it's very different. So this is why this is a very good analogy. And the whole idea is, instead of people interfering, it's chemicals interfering with one another. So the cells are just big sacks of chemicals, and keeping the certain chemicals separated so you can do different chemical reactions in different places and they not interfere with each other, that's very important. And it's very important to why we, as eukaryotes, were able to become multicellular, as in We're not just colonies of individuals. We are a single organism with very complex cells that talk to each other, and that communication is a very complex thing, and we wouldn't be able to do it if we did not have the organelles and this organization and all these genes, all this large amounts of DNA for storing lots of information, not only about our own cells, but how to communicate with the cells next to us, and allowing all the cells of our body to interact and form a single organism of many cells, and not just an individual colony of cells all acting independently. We act in unison to form a multicellular organism. Alright, and it's going to be very important for us to learn how these cells, to figure out what kind of cell they are. That's something called differentiation. We'll talk about that in later lectures towards the end of the semester, or no, that'll be towards the middle. But it's very important to know this story. This story is one that I always ask on the test in one way or another. So you do need to know the apartment and the mansion story, and why these compartments or these organelles are so important, and how lack of these limits the prokaryotes, the archaea, and the bacteria.